Okay. Welcome to the class. It is another day, another time, and it is just another class session. And it is for the first time I'm going to introduce this very new concept that I'm sure everybody is going to benefit from this. So mainly, I want to share some educational contents on biology, and specifically it is genetics. So I was trying to just write down some things there. So let's go to start it fresh. So mainly I want to share contents on what we call biology, and specifically it is from genetics. I'm sure at least everybody knows about this topic, genetics. So from genetics mainly I want to start with introduction that is going to show exactly how you are going to approach genetics questions, and specifically to have that connection between the questions and the concepts studied, because most of problems they are hard to be answered, or they become very tricky or difficult because you miss a link between what has been asked and from what you have been studied. So here I'm going to show that link. So specifically, let us see the concept of testicles. Testicles that I'm sure everybody have already heard about it. So always in testicles, what we start there is about the concept that we need to determine genotype of organism showing dominant phenotype. And because there is no way to measure genotype of an organism, there is no means. So basically, we only use testicles to determine that, and specifically for those organisms showing dominant phenotype, because organisms showing recessive phenotype, their genotypes are predictable. So we always deal with those showing a dominant phenotype. So mainly, what we do always in phenotype, maybe parental phenotype, we always close organism showing dominant versus organism showing recessive. So now organism showing dominant versus showing recessive, for example, in Mendelian experiment, Mendel was crossing what we call PEA plants, and mainly he concentrated mostly in characteristic of height maybe. So from that characteristic of height, now organisms showing dominant phenotype were those with the tall stems, and those showing recessive phenotype were those with the short stems. So mainly we can use it here, whereby now parental phenotype, now parental genotype. So the same thing on the phenotype. Now we will have those showing tall versus showing short. So now basically in the testicles, now always organism showing dominant phenotype can be homozygous. Maybe in the case of height, can be homozygous tall or can be heterozygous tall. And those showing recessive phenotype, always they have just a single form. They must be homozygous recessive. So basically, testicles is carried out to determine if that organism showing dominant phenotype is homozygous or is heterozygous. So that's the purpose of testicles. So let's go in. And mainly in genotype, therefore, organism showing dominant can be homozygous like this, and also can be heterozygous like this. So we are crossing it versus organism showing recessive phenotype that has got just a single form. So now mainly a cross can go as usual, but mainly a conclusion of a specific genotype of an organism is drawn from those results. What's going to be obtained there is the one that will give us a conclusion on the genotype. For example, Mendel, when he was crossing tall plants and short plants, now always when he obtained all resulting plants having tall stems, now the conclusion was his experimental organism was homozygous tall. I think we see this one. So if all the resulting individuals will have tall stems, now always this experimental organism has got this homozygous tall genotype. And now the alternative answer or the alternative conclusion was if all organisms that resulted from that clause were showing a mixture of tall and short. Now if he obtained a mixture of tall and short, now the conclusion was his experimental organism had this heterozygous genotype. So that's how you are going to draw out your conclusions on the idea of testicles about the genotype of organism showing dominant phenotype. So let us see, let's go deeper and see the cross. So now maybe let us consider this one, the first one. So always this will form gametes now during meiosis. Now during meiosis it will form gametes. Now we have caps T. Now this will be the gametes. Then this one also will form gametes, the same thing. Now after formation of gametes, now you label there as gametes. After formation of gametes, next is fertilization. Now you label there as fertilization. Now fertilization, it is fertilization. So there's F there. So now always this one will be caps T small t. Then this with the second one, caps T small t. This with the first one, caps T small t. This with the second one, caps T small t. So you will find that these are the genotypes of the offsprings. And now the phenotype will be, all of these will be tall in their phenotype. Now always in their phenotype all of them will be tall, but specifically in their genotype they will be heterozygous tall. But all in all we don't see the genotype, so our conclusion is primarily drawn from the phenotype. So that's how things goes. And now from there we can have the other one. Now what if, now let us see what if a genotype of experimental organism was this one, heterozygous. Now always we'll have parental phenotype. Now we'll have tall across the versus short. Now parental genotype, tall has got heterozygous form. Now always during meiosis, always they segregate caps T small t. Now this will be the gametes. Now the same thing, now will be the gametes. Now you can label there as gametes. Now next year is fertilization. Now during fertilization anyone can fertilize the other, caps T small t. Now caps T small t, this one small t. Then with the last one is small t. Now you'll find that we have those now two results, now two different results in the genotype. We have the other one there. So these are the genotypes. Then what will be the phenotypes? Now in the phenotype this will be tall and this will be short. So that's how we are going to draw the second conclusion. So having mixture, now you have across the tall stemmed plants and tall stemmed plant and short stemmed plant, then you are resulting plants, always they are mixture of tall and short. Some of them they are tall, some of them they are short, and always they are in a 50-50. We have a 50-50 ratio there, whereby we will have 2, then 2 there, 1 2 1 2 1 2. All of them they are over 4. Now over 4. So by 2 will be half, by 2 will be half. So we have half. Now this half. Now this means 50-50. It means if you have 6 plants now from your cross, so you'll find that 3 will have tall stem, 3 will have a short stem. So that's the general idea. There's no need to balance exactly 3 by 3, but always they range within a 50-50 range. So now this takes us to, when we simplify this now, simplifying this by half, we will have 1 by half, then we will have the other one there. So we have the 1:1. So always I think we have the 1:1, which is always used to present the test-cross ratio. It is known as the Mendelian classical test-cross ratio, and it is a ratio which is obtained when experimental organism always is heterozygous for the genotype. If experimental organism is homozygous, we'll have all of the resulting individuals showing dominant phenotype. So that's how you are going to do with this, and then later I'm going to link this with some questions, because I'll get some questions from test cross, so I'm going to link them direct with this concept. So thank you for your time.